The human genome harbors regions that have evolved at an astonishing pace since our ancestors diverged from chimpanzees. These are called human accelerated regions, HARs. But here's where it gets controversial. While many believe these rapidly changing sequences hold the key to what makes us uniquely human, the truth is far more complex. Miles Wilkinson, PhD, a leading researcher and distinguished professor at UC San Diego School of Medicine, shares a candid perspective. He suspects that over half of our genome is essentially junk DNA, and that many rapidly evolving sequences might simply be drifting without serving any specific function. What sets HARs apart, though, is that unlike most fast-evolving DNA, HARs are strikingly conserved across other species, hinting that they could be critical pieces in the puzzle of human evolution. Scientists have been fascinated by HARs for nearly 2 decades, identifying over 3,000 to date. Interestingly, about 90% of these lie outside the traditional gene coding regions, in areas previously dismissed as non-coding. This has made it challenging to pinpoint exactly what these regions do, especially in terms of traits unique to humans. Recently, a breakthrough came from the Wilkinson lab at UCSD with their detailed study of one specific sequence, HAR123. Their work, conducted using mouse models, is detailed in the journal Science Advances under the title "An Ancient Enhancer Rapidly Evolving in the Human Lineage Promotes Neural Development and Cognitive Flexibility." It's important to clarify, HAR123 is not a gene itself, but what researchers call a transcriptional enhancer. Enhancers are crucial regulatory sequences in our DNA that orchestrate when, where, and how strongly certain genes are turned on, especially during development. Wilkinson's team became interested in HAR123 through their research on the nonsense-mediated RNA decay, NMD pathway, a quality control system that helps reduce mistakes during gene expression, errors that can be particularly harmful to brain cells. Wilkinson explains that NMD is vital, not just during brain development, but also for the ongoing function of adult neurons. This led to their intriguing question: could any HARs be linked to genes involved in the NMD pathway that might explain human-specific brain functions? Their search revealed 3 such HARs, 2 of which, PAR53 and HAR123, are embedded in the SMG6 gene. The team zeroed in on HAR123 for functional studies. To unravel HAR123's role, the researchers used CRISPR gene editing technology to delete both copies of HAR123 in human embryonic stem cells. Their observations were striking. Without HAR123, these stem cells struggled to form neuroectoderm, the early developmental tissue that ultimately generates the brain's neurons and support cells called glia. Specifically, there was a noticeable decline in the expression of genes that mark neuroectodermal identity. Digging deeper, the team found that the absence of HAR123 led to defects in producing neural progenitor cells, NPCs, the precursors for neurons and glia. Additionally, mutant cells formed unusual clusters not seen in normal cells. The impact extended to the brain's hippocampus, a region essential for learning and memory, where the balance between neurons and glial cells was altered during both early and late postnatal development. But perhaps the most fascinating discovery relates to behavior. Mice lacking HAR123 appeared physically healthy and could complete various tests. Yet they struggled with cognitive flexibility, the ability to adapt and remember new information when circumstances change. This finding suggests that HAR123 plays a pivotal role, not just in brain structure, but also in the higher-order functions that underpin human thinking. Looking ahead, the Wilkinson lab is excited to explore HAR123 further. One ambitious next step is to replace the mouse version of HAR123 with the human sequence to see if it enhances intelligence or cognitive abilities in measurable ways. Such experiments could shed unprecedented light on what truly makes the human brain special. There is also a pressing clinical angle. The genetic area containing HAR123, located on chromosome 17, is linked to a variety of neurological disorders. Deletions or duplications in this region can cause a spectrum of symptoms, ranging from intellectual disability to seizures. This raises the tantalizing possibility that HAR123 might contribute to neurodevelopmental conditions such as autism. Additionally, the team is investigating if HAR123 is connected to diseases impacting the brain's white matter, like multiple sclerosis. However, so far, this link is purely correlational, and establishing a direct cause and effect relationship remains a goal. To accelerate progress, Wilkinson's group plans to collaborate with specialists focusing on these disorders to untangle HAR123's true role. This research opens exciting but challenging questions about the genetic underpinnings of human cognition and disease. Could an ancient, rapidly evolved DNA enhancer be a key driver of the flexibility and complexity of the human mind? Or are we ascribing too much importance to one small genomic region? Ooh. What do you think? Does HAR123 hold the secret to our unique brain power, or is the story more nuanced? Share your thoughts and join the conversation below.